is Dr. Jarek Meller. Dr. Meller is someone I've gotten to know really well over the last 4 or 5 years. He's at both the University of Cincinnati College of Medicine and Cincinnati Children's, and I believe has many, many roles there, which I'm not going to get into, but he has a very interesting background in physics, math, and computer science for his foundational degrees, and a PhD in computational chemistry from Nicholas Copernicus University in Poland, and we talked about this at dinner last night, an international tour of many, many countries for different postdocs, and we were very fortunate that he landed in the Midwestern United States. And just by way of introduction, there is a method to the madness for the sequence of the speakers today. So you got a taste of the links-based library of cell signatures analysis from Dr. Shuckle's talk. And Jarek will tell you about, I don't know if he's going to tell you about the structure of links, but he's one of the leaders in the development of this platform. And I don't want to say anything else, and I'll let him just take it away. But a lot of the link stuff that you've heard me talk about, if I've been around your departments, I got all from Dr. Meller. So, Jarek, take it away.
Thank you and good morning. I will probably try to walk here just to change the perspective for the next talk. It's about the same thing, but different perspective. So we just heard about somebody actively using links, and we are indeed part of the LINKS consortium in Cincinnati. There's something called Data Coordination Integration Center for the whole LINKS consortium. So Broad Institute and other places actually generate these massive data sets, and that all these data flow into Data Coordination Integration Center. That's really what I'd like to talk about and how we are using this to build tools to make all of that available. So it's not a research talk per se, it's more of a resource talk and also introduction to some of the things that will be discussed later, including by Jim Riegel, who's in the audience, and potentially Sonchai and Bukhur if they actually make it and still driving. But that's coming later in the afternoon session. All right, so Library of Integrated Network-Based Cellular Signatures. That's what the LINKS acronym stands for. And I would like to briefly show you how this can be used as a resource for pharmacogenomics and precision medicine, again with its extensions. And that will be covered later, including SIG2Leak and a little bit of PyNet and SWAP tools. But my primary tool for this presentation and also the first demo in the afternoon will be something called iLinks for integrated links. So this is again the overall structure of the consortium, and you can see that there's one place, maybe two places in between the coasts, including Ohio. So we're certainly proud to be part of this, and the whole Data Coordination Integration Center actually consists of 3 centers, really, Mount Sinai, University of Cincinnati, and University of Miami. And you see the data generation centers, including Neuralinks, with several centers involved. And it was mentioned that it all started with connectivity map concept. Just wanted to remind everyone that that comes from Todd Golub and his group. That's the whole purpose of this slide. And the whole setup was already described, so very briefly. The whole concept consists of building a library of signatures, transcriptional, and now increasingly also other types of signatures of perturbations. So those perturbations could be small molecules, but there are actually also genetic perturbations, as was mentioned already. And there are 2 types, too many types, I will come back to that, actually important for what we do. And in each instance, an experiment is performed to identify differentially expressed genes with respect to some baseline, and that defines these upregulated, downregulated genes in response to a given perturbation. For any particular system of interest, that could be disease state, that could be some model system, that could be some particular perturbation considered in a lab. One could generate obviously a signature, and then compare that signature with a library. And assuming that we have a comprehensive library and we're getting there, we can potentially find signatures in the library that is either strong positive or strong negative correlation. So these will be strongly concordant, those will be strongly discordant. So we can think of, if we have discordant signatures of this drug, potentially reversing signature of a disease state if this is what we are measuring. So that's the overall setup. You can see examples of actually using this concept to find drugs that may, for example, reverse the drug signature. And first one is to identify drug resistance signature, and this is in the context of leukemias. One can identify cells with resistance, compare those with cells without resistance, identify signature of resistance, and find drugs that reverse that, and you can see that in this particular case, rapamycin and mTOR inhibitor turn out to be an interesting target. By the way, you see these kinds of plots that we just explained.
Yeah,
I have an
thanks.
example here. All right, so with those kinds of examples, the overall promise, which may be a little bit high, but it's certainly there, is that this could be used as a resource for personalized precision medicine, following this overall setup. So we would generate maybe transcriptional signature of a tumor, particular tumor, and would hopefully be able to find molecules that reverse that signature, and then test first on cell lines, and then potentially move, if these are already, and many of the small compounds considered by links are FDA-approved drugs. This could be used directly maybe for treatment, at least in a more sort of experimental setting. But this goes way beyond that kind of promise of personalized precision medicine. There are all kinds of things that can be done, including examples of what we have just heard about, with model the action, identification by comparing similarity between signatures, but also insights into loss and gain of function, potential drugability, because we have gene knockdown and overexpression, gene overexpression signatures. So loss and gain of function are being captured, and we can see how those are related to the loss or gain of function for other genes, how those are related to drug signatures, and what could be connected. So I guess that goes back to the original name, connectivity analysis. And of course, you can use this for overcoming drug resistance, drug repositioning, and so on. All of that is dependent on the large data collection, and that's the overall LINKS consortium, but also on building tools and platforms for accessing and interacting with these data. So in terms of the overall growth, just to summarize this, looking at the what we call LINKS data cube, one dimension is cell lines, and touchstone subsets of most systematically performed large-scale screening. That's only 9 cell lines, but overall there's hundreds of them, and data for many of them gets quite sparse, but that's one dimension. Then perturbations, that's all the genetic perturbations, all the chemical perturbations, and so on. So you see the scale, 10 to the 5th, maybe perturbations. And then all kinds of assays that L1000 is just one of them, in fact, because LINKS grows in all 3 dimensions. So we have more cells, more assays, including proteomics, including those P100 and GCP targeted proteomics assays, and more perturbations, including CRISPR in particular as of late. So the initial gene knockdowns, we should probably add to this, were mostly done by shRNA. But as of late, this is increasingly CRISPR. So this is more or less where we are right now. And again, we saw snippets of that. So the 42,000 small molecules is a little misleading. We have a lot of data really for 20+ thousand. And some of the numbers with this number of cells is also quite misleading in some sense, because for many of them, we have very sort of limited sparse sampling. Again, some of the rich data set are done in much smaller number of cell lines, and these are primarily cancer cell lines. But as you could see previously, we also have some differentiated neurons and other brain-related cell lines too. All right, so all of that is available. Whatever is generated within a couple of months of processing, making sure that it's all harmonized, sprinkled with metadata, and so on and so forth, is made available. And one of the places, and I will be showing this briefly in the demo later, is Nick's data portal. You can just go there and download everything at different levels. Higher level data, process signatures, and also lower level data. You can also download Dockerized pipelines that take the lower level data to the signatures. They can play with those pipelines yourself, change parameters if you like, validate them yourself, and so on. But it's sort of work, and it takes a lot of time to develop any sort of in-house solutions to be able to deal with that. So in addition to that, we are making sure that you can interact with these data more directly. And one of those efforts actually comes from Mario Vedovich's group in Cincinnati. We're part of that too, but this is very good. So I want to make sure that everyone remembers that. And that's called ILINKS, for integrated links. And one of the things that ILINKS does very nicely, and there are other resources like ClueIO developed by Bro and so on, but what ILINKS focuses on is to actually perform on the fly, or even pre-compute in many instances, connectivity analysis, where you consider two main types of perturbations. One is genetic perturbation, primarily by the siRNA, but also by CRISPR, and the other is chemical perturbation. So we can either inhibit function, we can have loss of function due to protein inhibition, protein product inhibition, or due to gene knockdown. Those will have different types of off-target effects. One sequence-driven, the other structure-driven. So that alone allows for the more careful validation of what's off-target, what's on-target. But you can also find correlations. By finding correlations between these signatures, and by the way, these boxes here represent genes. So you have gene 1, gene 2, gene 3, 4, 5, and 6. Very small signature. The blue is down-regulated, yellow is up-regulated. So you see that in response to gene knockdown of some gene, this gene goes down, this gene goes up, and so on. And we have an inhibitor, and you have some similarity to the level of signature. So we might have a gene in a pathway that inhibits the entire pathway, and if we have a specific inhibitor of one of the pathway members, and the downstream echo, transcriptional echo, could be very similar. We can detect that those two things are connected. And again, you can go back to the original data, or you can do this with tools like ILINKS. So I have a quick offline demo, just to give you a sense of how easy this is with tools like that. And I need somebody to remind that I'm running out of time. I'm probably already running out of time.
You're doing great.
So just let me know when I really need to stop.
You got about 9 minutes.
So if you go to ILINKS, things keep changing. We just reorganized a little bit. The interface of signatures are going to be your first choice. So if you go to signature workflow, there are different workflows in ILINKS, and I will present some of them in my demo session. You can type gene name of interest, you can type drug name of interest. Chances are it's already there. And a big caveat here, there are 4,200 or so, if I remember correctly, gene knockdowns, and around 2,000 gene overexpression experiments done in some cell lines. That's not 20,000. It's way less than 20,000. But it also goes sort of a druggable subset of the genome. It wasn't a random choice. Those were the first ones to be tested. So chances are that your gene is not there, but if you're interested in some druggable, you can look in the neighborhood of that gene. Actually, we have other tools that drive that search that way. So you start with the gene. If it is not there, it will show you network neighborhood of that, and it will show you which of those things are available. But if you just type something like this on, which I know is there in the signature workflow, it will retrieve anything related to the query, which is hopefully a set of gene knockdown signatures for estrogen receptor alpha. And indeed, we get a whole bunch of them, and they are displayed in the form of these kinds of tables initially. And you see that there are multiple cell lines. You can recognize those microbiologies that these are cancer cell lines where knockdown of this gene using shRNA is performed. Actually, typically there was maybe 9 shRNA, different shRNAs, and then the consensus signature is derived from those 9 experiments in order to minimize the off-target effect. As you know, shRNA, even CRISPR is not perfect. So we can take one of those signatures and look at each one of them. They each have a landing page, so you can click on that. This is MCF7, breast cancer cell line, and it will tell you all kinds of things. You can analyze what's in the signature, what genes go up, what genes go down, what pathways it represents, and so on and so forth. All kinds of, just in terms of analyzing what kind of biology is associated with the loss of function for this particular gene. But the critical thing that it also does is these connected signatures for gene knockdown signatures and for chemical perturbations in particular. They're all other, I-links covers much more, and actually, in addition to links data, it covers systematically derived signatures from geo data sets. So that's also a great resource, which is there. TCGA is there, many other data sets are there. So these analyses, they go across different databases, if you will, and these type of consortia. So, but if we stick to links, we can look at chemical perturbation signatures that are connected, concordant or discordant, to the signature of estrogen receptor alpha loss of function induced by shRNA. And if we do that, just click, it's already there. Sometimes you need to also compute something additional to refine that search, but there are pre-computed results. And if you click to recompute, it doesn't take too long, and we're working to improve it further. And you see that, for example, things like raloxifene, which is a non-inhibitor, are positively and in statistically significant manner, these are concordance scores, correlating with the loss of function due to shRNA. So we have this kind of, this is a well-known example, that's what I'm using here. So we can induce loss of function just by knocking down the gene, or if the gene is still there and makes the product, we can use chemical inhibitor, small molecule, to block its function. Obviously, again, the off-target effect and overall effect on biology will be somewhat different, and that's why these correlations are far from perfect, but they're still significant. So if you look in the other direction, negatively correlated ones, you see estradiol. Estradiol is an agonist, something that is required for function. So loss of function goes in the opposite direction to gain of function, and that's what we would expect. That's kind of a nice example for this kind of presentation. I'll have some more examples later. And before I switch gears, to motivate what I would like to just say as a teaser to the next talk is that when we look at the pathways, it gets, of course, potentially quite complicated because pathways are complicated. But even if we assume simple linear pathways, even in that case, we have to be careful because if we have signaling cascade, disrupting a cascade can result in very similar downstream effect. And whether we disrupt this at higher level or lower level, so that adds to the noise, and then you have all kinds of off-target effects. So these correlations that we see here, for example, between PIK3 knockdown in some cell line, but this is MCF7, and no inhibitors of PIK3 kinase, all these drugs are non-inhibitors, and we have their signatures in links, and we can compare this with signatures of a knockdown of PI3K. You can see that these correlations, again, on some of them are actually suggested to be significant, some are not. They are not really all that strong. So that leads us to SIG2 which is what Jim will tell you more about, but I wanted to just briefly mention because it flows with the overall sort of philosophy of what we are trying to do. Because on the one hand, our big task is to simply make sure that all of you are happy by being able to download data in harmonized, annotated form of rich metadata, and that you can interact with the data. But as part of that interaction, in order to bring this closer to biology, learning from what we are doing, we're trying to develop better tools. And one of those tools still under development is SIG2 from signature to lead compound, where we're actually combining this kind of signature connectivity analysis with atomistic simulations, molecular simulations. And the reason for doing this, again, it's level of noise and relatively weak signals that we often get. And if you consider a pathway, it's again idealized, almost ridiculously simple pathway from EGFR to subkinase to June. So from the membrane to the transcription, so to speak, pictorially, really, inhibitors targeting each one of those targets will generate signatures. And so will knockdown of any one of those. So if we are after inhibition of SRC kinase, because that gives us some advantage, some more specificity, if we take knockdown of SRC, all of those three will be somewhat correlated to this in the simplified picture. Upper genes down and upregulated will be similar to down and upregulated here. And one way to deal with that is to combine signature concordance with target knockdown, with shape complementarity. So we now combining two principles. Docking simulations can be used to take those small molecules that are candidates by virtue of having some concordance with gene knockdown for a target that we actually know and we want to inhibit, and we have a structure of any kind. But if we do, we can run docking simulations, and we have two noisy ranked lists of candidates, and by combining them, we can get better results. And that's really the essence of SIG2, the overall principle behind this. There are other tools that we're developing, including this platform for proteomics data analysis, which is PTN-centric, post-translational modification-centric, and it can generate these kinds of kinase network views where you have site-specific information. This comes from some abundance profile. Oops. Too quick, sorry. This comes from some abundance profiles for some PTM sites and some proteins. These are kinases that are either known or predicted to target those specific sites. So if you have something going on in some experiment and you see the kinases that are specifically phosphorylating that site, that phosphosites, that could be how you interpret the results again by identifying kinases that may be involved. There are many other ways to do it, but we're trying to make this as simple as possible. And these are the people who are actually doing the work. And so that's Alex, Jim, who's here, Cruz, Guillaume, Samchai, and other collaborators. And with that, I would like to stop, and I would be happy to take some questions.
That was a nice talk. You guys hear me? Yeah. So it's a nice talk. So the problem with reproducibility in science, right? There's a epidemic of not being able to reproduce results. How does iLinks control the quality of the data that they're putting on in their system?
Enormous effort goes into actually making sure that we do as much as we can to not only document everything that happens, to the point of providing Dockerized pipelines for anything that happens, so that you can download those Docker configuration files and run it yourself, but also to benchmark every step of the way. And that, much of that, I should also say, is done at the data generation centers like Broad. The quality of their proteomics data, for example, is just amazing. Because there's so much effort going into this. Everything comes with caveats, and I should thank you for this question because I have this on one slide. And we are dealing with noisy biological, stochastic biological systems with noisy readouts. And another big caveat, if I say, is that we are working with cell lines, and the question is how that extrapolates to real life biology. That's another big caveat. But for iron specifically, even simple things, quote unquote, like measuring correlations, there's at least 2 years of benchmarking of what is the optimal way of deriving a concordance measure for signatures. We do have certain things at our disposal, for example, technical verifications. And these are not trivial technical verifications, but Broad does for L1000 is that every once in a while they will redo something on a different plate. So we have tons of those. We have same drugs, but different cell lines, different concentrations, different times of exposures, and so on and so forth, and again, different experiments. We then posit that drugs with similar mode of action should, again, cluster better than drugs, different modes of action, and so on and so forth. So we use all of those things to analyze carefully what is the best way of measuring even simple things like correlation between signatures, and it goes from there every step of the way. So we're making an utmost effort to make sure that everything is documented. Speaking of which, Eiling's paper is in the final stages, so that you can read more about this hopefully soon. INET paper is in the final stages. Sig2Lead paper is in sort of final stages. So none of that has been published for those particular tools that I mentioned, that to be fair, and some of it read about once it's published, but we are certainly making an effort to minimize those.
But we have time for another question. Okay.
Thank you so much. I'm going to try to summarize 3 questions into one. So if I'm submitting an R1 and I want to incorporate this as a tool for showing innovation, a new way to what are the reviewers going to say when I say I drank the Kool-Aid and I've done this? If I'm going after a cardiovascular phenotype, are they going to say this isn't the data set for you, you should be using something else?
Actually, Mount Sinai has a whole center dealing with cardiomyopathies and other cardiac phenotypes, and they're doing this in their cell lines. So there's plenty of data to do. There's this broad effort to get as much data as possible, mostly on those 9 cancer cell lines. But in addition to that, there are these more focused efforts. They may not be as broad, but are much deeper and much more focused. So I will go with that, and you can use that to validate some of the findings, potential hypotheses from a sort of broader search.
Thank you so much.
One more question up front.
Yeah, you talked about the rapamycin reversing the glucocorticoid signature.
Yeah, that blocked my work. I hope this was clear.
You talked about rapamycin reversing the glucocorticoid signature. So my question is, when would you call that as a reversal? What percentage of the genes in the signature should be reversed? 50%, 60%, or a total reversal? Because it doesn't always happen, 100%.
Yeah, so I wouldn't necessarily quantify this in terms of number of genes because of the nature of the signature that we're dealing with. So we have these landmark genes, 978 landmark genes, that were selected to be representative on different pathways, and also as a basis, a proxy for other genes, so expression of other genes could be imputed from those. So because we have 978, and it's already built with sort of data science concept in mind, it doesn't necessarily represent in a comprehensive way the order of biology. It's good as a signature, but not necessarily to count genes actually. What we can do if we have multiple signatures with 978 is to ask what is the range in one signature versus the other? How far from a mean expression they go up or down? And measure correlations. So we would quantify this mostly in terms of the strength correlation. So we want to see extreme behavior that is very concordant or discordant. So the genes around the mean would not contribute much, and genes that are farther removed from the mean behavior will contribute more. Actually, we are even emphasizing this more by using extreme correlation rather than standard correlation.
For example, can you call if the efflex drug transporters, they're up, right? There's drug resistance. If they're down, can you call it reversal?
Again, we have 978 landmark genes. We know that have more than one or two of those.
So this is an interesting discussion because this has come up in our lab meetings a lot. And what I've learned from the last 5 years with Dr. Meller is signature-based analyses are very different than having the candidate gene and chasing your A to B to C pathway. And so there's a richness to signature-based analyses that is agnostic to your a priori hypothesis that this is the gene that's involved and I'm going to chase this transporter or that transporter. Because looking at 1 or 2 or 3 or 4 proteins can completely mislead you.
But if I may add one more comment to that, if you are suspecting some transporter and that transporter has already been knocked down and you have signature of loss of function for that transporter, that you can try to connect this with itself because now you're in the same space of 970 readouts that define where the response is similar or not.
We'll do one more question.
Okay.
Thank you. What will happen if your protein of interest doesn't match with the island database?
Yes, so ILINX, if it doesn't find something, it will always tell you that. But we have other tools that are built around the concept of sort of fast into full match because we have gene, cell line concentration, or compound, cell line concentration, and so on. And so we're trying to find the closest matching in the database. So we have another genes proteomics that starts with that kind of search and will tell you whether there's something related in the pathway or something related due to some other annotations that could be started. Some of the fastest search will be implemented in ILINX too.
So we're gonna keep moving. Thank you, Dr.